and welcome to the Brain and Transcranial Photobiomodulation Virtual Summit. I'm Dr. Joe DeDero, your host. My guest today is Dr. Jerry Ann Lyons. She's the Associate Dean at College of Health Sciences at the University of Wisconsin-Milwaukee. She's a Professor of Biomedical Sciences at the University of Wisconsin-Milwaukee. She's internationally recognized for her work in photobiomodulation for MS, multiple sclerosis, and autoimmunity. She's an expert in the mechanisms of photobiomodulation, and her and her team have completed the first study for the treatment of multiple sclerosis, MS, with photobiomodulation in human patients. So thank you so much for being here, Doc, Professor. And let me start off with asking you, most of the researchers here have spent a lifetime researching. This is their life's work doing research, but how did you get started with your research in photobiomodulation and sort of how that led to the brain in your work there?
Sure. So my research is always focused on multiple sclerosis and the role of the immune response in multiple sclerosis. And in 2003, I started as an assistant professor at UW-Milwaukee at the same time that Dr. Janice Ells started at the university. And so for the first year or two, she would tell me about photobiomodulation and how she thought it worked. And I would say, well, if what you say is true, then photobiomodulation should make my mice feel better. And after a couple of years, we got down to doing the experiment. And it was something that I never thought would work. I never expected it to work. We did the first experiment, and the experiments in the mouse model for MS take a good month, if not two, depending on how they're set up. And so I'm watching the mice through the whole period, and I'm thinking, yeah, this isn't working. If anything, I thought maybe they were worse. And so the end of the experiment comes. It's late on a Friday, because when you're a research scientist, what else would you be doing late on a Friday? I sat down at the computer to analyze the data, and once I did everything I needed to do, I sat back and looked at the graph, and I'm like, well, by gosh, it worked. Surprised the heck out of me. And so I immediately called Janice, and I said, you're not going to believe this. It worked. She says, well, of course it did. And so now that was 2006, and so now for the last 14 years, it's been the main focus of research in my lab.
Wow. I've had that same story from almost every single researcher that I've interviewed. I think it's the drive. It's not the drive to know, because they already make up their mind that it's not going to work, and then that's a perfect way to do science, though. So, think about that, 16 plus years of effort, and you're gonna bring that to bear on us today. I can't wait. Go ahead, Jerry.
Okay, sounds good. So today I'd like to explain a bit about photobiomodulation. What is photobiomodulation? The parameters that we need to consider when we consider dose. How do we define dose for photobiomodulation? And then finally, what are some of the biological effects of photobiomodulation? So quite simply, photobiomodulation are the biochemical and functional changes in cell function induced by the exposure to light at certain wavelengths and at certain doses. Those 2 points are very important, and we'll come back to those. So the important thing to keep in mind is what we're looking at here is light initiating a biological response. This is not unheard of. We all are familiar with photosynthesis, for example. We're familiar with vision and how vision works with rhodopsin. So quite simply, the cells absorb light, and then there's a molecule in the cell termed a photoacceptor, which will absorb that light, and then based on what that molecule is, what its function is, different biological processes are initiated. And we'll go through these in a bit more detail, but these can be biochemical reactions, signal transduction pathways, and most importantly, leading to changes in protein expression that affect the health and well-being or function of the system itself. So if we consider biological effects of light, we know that these biological effects can occur across the spectrum. We're all well familiar with the role of ultraviolet light in the synthesis of vitamin D. Without sunlight, without ultraviolet light, the body cannot synthesize vitamin D. But on the other hand, ultraviolet light is also mutagenic. It will damage our DNA. It will lead to cancer and other diseases if we are exposed to too much ultraviolet light. We know that blue light now is antibacterial, quite effectively so. But we also know as we use our electronic devices that it is damaging to the eyes and can lead to eye strain. But we also know that it's important to regulating circadian rhythms, or the basic rhythms that allow the body to function based on the time of day, based on what is necessary. As you can see, green light, yellow light also have important biological functions. Yellow light is a little bit, I don't want to say controversial. We know a little less about what yellow light will do, but we do know that it's quite effective in cosmetic applications, reducing wrinkles and that sort of thing. Today, we're going to be focusing on red or near infrared light, specifically light in the wavelengths of 600 to 1000 nanometers. And we're gaining a lot of knowledge in the past number of years. We know that red light is anti-inflammatory. We know that it can induce vasodilation or expansion of blood vessels that allows blood to flow more freely through a site. It also increases cell proliferation, which is important when we're talking about activation of the immune response and the protection against disease, as well as wound healing. Photobiomodulation has been very effective in wound healing, and it's in part through its effects on cell proliferation. And then also important to a number of our applications is it actually protects against cell death, which is a natural consequence of aging, but also disease. So just a few definitions to get us all on the same page. Photobiomodulation itself is the process by which a chain of biochemical reactions is triggered by exposure to light. Photobiomodulation therapy, on the other hand, is the therapeutic application of light to elicit a biological response. So related, one is the process itself, one is the therapeutic application of that light. And quite simply, photons are absorbed by, as I said, a photoacceptor molecule, ultimately leading to changes in gene transcription, or activation rather, and biological responses. Now, the wavelength of light that is effective is defined by what we term the optical window, which is quite simply the wavelengths of light that the body is able to absorb, that the molecules are able to absorb to elicit the response. So when we consider the mechanisms of photobiomodulation with red light, the important thing is the photoactive part of the cell is the mitochondria. And the mitochondria are very important in cellular function. They're responsible for producing energy in the form of ATP. Without energy, our cells can't perform their biological responses that keep us all healthy and disease-free. But it's also the major site of the generation of oxidative stress or nitrosative stress, ROS, reactive oxygen species, or reactive nitrogen species, which are a balance between beneficial effects and detrimental effects, which we'll talk about momentarily. But it's through this production of reactive oxygen and reactive nitrogen species, the generation of ATP, that the mitochondria are really central in then regulating the life and the death of the cell. Think of the mitochondria as somewhat of a barometer. They're able to, or they sense when things are going well, and they keep doing what they're doing, but they also sense when things are not going well, and they will set the process of cell death they will start that process if it's necessary. excuse me. Now, if we take a little closer look, this is actually a micrograph of a mitochondria. You can see it's quite a complex little organelle. These are inside the cell. We think that evolutionarily they are actually originated from bacteria that the cells engulfed, that then were able to evolve to help us. You see a lot of membranous structures. All these little areas penetrating into the mitochondria are cristae, and it is along these membranous organelles that the processes that we're about to talk about take place.
Not the outer membrane of the mitochondria, the inner.
The inner membrane, right. So if we look in more of a cartoon format, what we see is it's the mitochondria have their outer membrane, which defines their structure, their outer appearance in the cell. But all of these cristae are really inner membrane that that folds in, and we can see that within each of these membranous structures, we have mitochondrial DNA. This is important to generation of ATP and such, also important to what we see with photobiomodulation. But it's within this inner membrane space that the processes of electron transport take place, that the effects of photobiomodulation are originated. Now, this looks complicated. I'll make it as simple as possible, but this is really the crux of where photobiomodulation starts. And so we're looking at that inner membrane space. This is where ATP is generated, and you see these different protein complexes embedded into. Here is our inner membrane. This is our outer membrane, inner membrane space, and the matrix, as you just saw in our last figure and also up here. And so what happens is these, there's 5 complexes. One is entirely embedded into the cell membrane, but to generate ATP, we move electrons from quite originally labeled complex 1, complex 2, 3 is inner membrane, 4 and 5. And so we pass the electrons from each of these complexes down. This is our complex 4. This is our cytochrome oxidase. As we generate, or as we transport electrons through each of these through each of these complexes, in order to produce that energy, we're pumping protons into the inner membrane space here, yep. And this then provides us the energy. And so as we get to cytochrome oxidase, or cytochrome C oxidase then is going to take that electron, transport it to our cytochrome 5, or our complex 5 or ATP synthase. And as we do this, we pump a proton into the cell. That gives us our energy, and it's that passage that then makes the ATP molecule that is the cellular energy. So the cytochrome C oxidase is the enzyme that transports the electron, and it is that molecule that is absorbing the light and affecting mitochondrial function, initiating the processes of photobiomodulation.
I like that. Now, can I just go one? So this is a great explanation and bringing it down to us now. If that ATP now is within the inner membrane of the mitochondria, and we've pumped back and forth our hydrogen ions. Now, how does that ATP energy get from the inside of the mitochondria to the cell that might need the energy? Because the cell function, the nucleus of the cell and what have you, that uses the energy too.
Sure. So let me take a step back and first say that this leakage of protons into the inner membrane space And this process of ATP generation does produce a little bit of oxidative stress. But this little bit of oxidative stress, which we'll talk about momentarily, is beneficial. It helps the cell signal. It has other functions to maintain.
Right.
Physiology, when this process goes awry in conditions of disease or stress, we generate more oxidative stress, more nitrosative stress than the cell can handle. And it's that process that photobiomodulation helps to correct. Now, once this ATP is generated, there are mechanisms in the cell that basically transport it to where it needs to be so that the enzymes in the cytoplasm can do their thing, enzymes in the nucleus can do their thing. Some of it is used by the mitochondria itself. That is all the energy that takes all of these enzymes to function also. So this is the primary site of ATP generation in the cell, but then that ATP is distributed through the cell so that the whole cell can function.
The bottom line is that this is you'll show us how this is gonna give us the more power.
Yes, yes. So when we talk about reactive oxygen or reactive nitrogen species, I've already alluded to the fact that these are beneficial, but they're also detrimental when they reach too high of a concentration. So physiologically, the ROS, RNS are important for signal transduction. This is the process by which the cell can detect one event happening and communicate it to another part of the cell so a biological response can occur. It's important for regulating cell death. One of the main signals for cell death to occur is when we have too much ROS or RNS, and the cell then it is triggered to die. Vasodilation is very important for regulating circulation. Hypertension or high blood pressure often is constriction of the blood vessels. And so vasodilation helps open up that blood flow, helps keep the body oxygenated. In terms of wound healing, it also helps transport the immune response and remove debris from the site. Reactive oxygen, reactive nitrogen species are critical for the immune response and for host defense. The oxidative burst is a process used by two populations of immune cells. That is the first line of defense against bacteria, for example. And so without this generation of ROS or RNS, we wouldn't be able to defend ourselves effectively against bacteria. The flip side of that, though, is it's also important for the downregulation of the immune response. And when not present, the immune response becomes overactive. It can lead to chronic inflammation. It can lead to autoimmunity. And so very important for regulating that. But when we override the mechanisms that control the generation of reactive oxygen or reactive nitrogen species, then they go from being beneficial to being damaging. They modify the lipids in the cell. They modify the proteins, the DNA. And the problem is once these molecules are modified, they're no longer biologically active and they need to be removed. So just as too much oxidative stress is sensed by the cell, its accumulation through the modification of our macromolecules is also a signal to the cell that something is wrong, and is a signal for death. We know now that many of the consequences, if you will, of aging are due to the accumulation of oxidative stress in the body, and is a main trigger for many of the diseases of aging that we see. And we're learning more and more that oxidative stress is at the basis of the pathogenesis of a number of diseases.
Now, I've seen you do this before, and the point that really has been resonating with me recently is this immune response and the oxidative burst. First of all, to talk about the pros, you know, they talked a lot about how a peroxide burst is what kills the bacteria, or a virus. And when we're looking at our immune response, you've kind of elucidated on this, and not too, because you're the immune system boss lady, that no one's kind of been able to add this to the whole system. That when you're looking at any tissue, you need to be able to defend yourself, and brain tissue in the same way. Mm-hmm. So these bursts that are generated because you have more energy, and I don't really know how this happens, how a cell can, the immune cell can go zip, bam, and kill the other cell with a blast of whatever it does. So that's the first thing I want to know if you can talk about. And then the second thing would be that having too many sick cells is what makes you sick, in one sense. So if you don't have the ability to kill the apoptosis, you can't recognize that this is a bad cell or a sick cell or a weak cell, then you're having more sick cells. So you have to build your health. And this is one of the major processes that photobiomodulation contributes to, is that hormesis. Hormesis. Stress the cell, and if it's weak, it's going to die. So it's kind of this is why you have pros and cons, because it is accelerating or amplifying a natural process. Would you want to follow up on that?
Sure. So, okay, so first of all, apoptosis is the death of our cells.
Cells.
Yes. So yeah, that's so when our cells die, there's a lot of different ways, but there are two main ways that cells can die. One is by necrosis. One is by apoptosis. Think of necrosis as an exploding cell, because that's really what it is. Necrosis is a very damaging response. Your cells are basically exploding and releasing all of their contents into the microenvironment of the body. And those themselves, so what happens then is when these cells die by necrosis and are releasing all of their contents into the tissue, so let me take a step back, because it's important to your other question. So the immune response has what we term innate immunity and specific immunity, or adaptive immunity. And the macrophages and the neutrophils are part of the innate immune response. They are the first cells there when some signal is received that either we have an infection that needs to be controlled, or we have too much necrotic cell death and they have to come in and clean up the mess. So the macrophages and the neutrophils will come in and they'll clean up that cellular debris, just as if they'd come in to try to kill a bacteria. But that's a very pro-inflammatory process. And we'll touch upon this. Inflammation in and of itself is not a bad thing. We need a little bit of inflammation to clean up just the general cell turnover that we have every day. We need inflammation as a first line of defense against infection. But when it becomes chronic, it's not able to be downregulated. And that's the problem with necrotic cell death, is if it continues on and the macrophages are there trying to clean up the mess, well, if the mess never goes, neither do the macrophages, neither does the inflammation. And then that sets up the chronic inflammation that will lead to a chronic wound. For example, a diabetic wound ulcer or a pressure ulcer is basically due to an overactive immune response who's there trying to do their job, but basically the stimulus isn't removed, and so they can't go away. Now, apoptosis, on the other hand, is more of an implosion of the cell. It basically just kind of involutes into itself, and we don't release all of that inflammatory cellular debris into the tissue. And so that part of the healing process doesn't need to happen. The macrophages don't have to come in necessarily to clean it up because nothing's being released to be cleaned up. Okay, so it's a much more controlled death. It's a much kinder death. to the system. Now, if on the other hand, the macrophages are there because of bacteria, there are the macrophages, the neutrophils, express different molecules on their cell surface that are able to detect or sense these bacteria or viruses that are present. And when we talk about the generation of reactive oxygen or reactive nitrogen species, and in particular nitrogen species at this point, there are three main enzymes that do that. eNOS, or endothelial NOS, is in the endothelial cells of the blood vessel, and it is largely responsible for producing just a little bit of nitric oxide, and it is that nitric oxide that's responsible for vasodilation, for example. In our neurons, we have nNOS, or neuronal NOS. And this enzyme is responsible for producing just a little bit of nitric oxide that's necessary for signal transduction, or for nerve transduction, I should say. Now, the third enzyme is called iNOS, or inducible nitric oxide synthase. And this is the enzyme that's present in the immune response in macrophages that's responsible for this oxidative burst. And the difference, one of the main differences between iNOS and the other forms of NOS is, first of all, it needs to be turned on. And that's some of those signals that the cell gets because the bacteria is present. And it produces huge amounts, relatively huge amounts of nitric oxide. And it's that large production of nitric oxide. So just as too much nitric oxide is going to damage our lipids, our protein, our DNA, the flip side is too much nitric oxide is going to do the exact same thing to the bacteria. Right, exactly. And so if all of this damage in our own cells leads our own cells to die, the same thing is true in the bacteria. We are modifying all of their lipids, all of their proteins, all of their DNA to a point that they can't function. Their function is compromised. And then between the macrophages and the adaptive immune response, we can go then in, clean up that mess,
Perfect.
Remove the bacteria, and the body recovers.
And I know that people that are watching this may seem a little bit like on the outside of their comprehension, but if we turn it inward just a little bit, what we're talking about here is the pandemic, that people are now saying we have too much inflammation in the body. We have this overreactive immune system that just keeps going and going and going, and that creates all this trouble, and that the nitric oxide supplementation, or people that are having higher levels of nitric oxide, are doing better, specifically for the reason that you just explained. Am I correct?
Well, to a certain extent, yes, you are. I mean, the immune response is in there trying to clear the virus, the coronavirus. The problem happens when the coronavirus gets into the lungs. And you might want to mark this. I'm not sure you're going to want to spend a lot of time on this. But what happens in the lungs is the pneumonia that develops is immune infiltration and generation of that immune response, but the immune response becomes too active and we generate what's called a cytokine storm. And the cytokine storm is basically the immune response in overdrive. It's working so hard to try to clear the virus that our normal tissue is collateral damage. And there's so much inflammation, there's so many cytokines being produced to combat the virus that our own cells are damaged as well. And that's where a lot of that's where the lung damage with coronavirus comes from, is an overactive immune response. Basically, yeah, it's the immune response out of control.
And that's what and so because if it had a stronger it's an inflammatory response because the immune system cannot mount and kill them quickly.
Well, right back. So the virus is really very sneaky, and it does its best to hide from the immune response. And so with the way the coronavirus works, it kind of sets up shop first in our throat, and really starts to override systems there. And then by the time it makes it to the lungs, it's evaded the immune response to a certain extent where it's harder to combat it. And so when the immune response makes it into the lungs to try to control the virus already has a leg up. And so it's a little bit of a catch up trying to control the virus. And in that effort, the immune response responds in a certain way almost too dramatically. And then the cytokine storm, as it's trying to combat the virus, is what's damaging the lung tissue.
Thank you for that. I just wanted to touch on that.
Yeah, sure.
It might come up. Some people are going to be talking about that as we go forward, because everybody's talking about it. And it's already on the top of people's minds.
Well, of course. And actually, I have seen a few discussions about what about photobiomodulation for coronavirus. And as an immunologist and knowing what's happening there with the cytokine storm, I would proceed with caution. We know a lot of how photobiomodulation affects the immune response, but we're still learning about the immune response to coronavirus. And understanding that balance is going to be really important when we start trying to treat it.
If you start popping off as we go down here in some of the list of things that we do, I mean, a reduction of cytokines has been seen, is what I mentioned. I mean, we've been able to reduce the cytokines and increase.
Well, you reduce some cytokines, you increase others. It's not an all or nothing proposition.
There you go.
So.
There you go.
All right.
Proceed.
What's next? All right. Cytochrome C oxidase. We'll go through this pretty quickly. This again is the molecule in the mitochondria that's responsible for absorbing the light and setting the whole cascade of events into action. It is complex 4 of the electron transport chain. And is really a key regulator of both mitochondrial function and cellular function. Now, what's important to our discussion is inside the cytochrome C oxidase is basically a toggle switch. And if oxygen is bound to that toggle switch, cytochrome C oxidase is functional and we're making ATP. Everything is happy. If, on the other hand, nitric oxide is bound to that toggle switch, the cytochrome C oxidase is turned off, and we're not generating ATP. And so in a normal physiological response, or in normal cell function, I should say, it's a balance between how much oxygen is bound to and take a step back. The important thing is there's not just one cytochrome C oxidase molecule in a cell, or just one cytochrome C oxidase in a mitochondria for that matter. And so we get this regulation of ATP generation based on how many cytochrome C oxidase molecules are bound by oxygen and turned on versus how many cytochrome C oxidase molecules are bound by nitric oxide and turned off. And so when the cell is functioning appropriately, this balance is good. We've got oxygen bound to enough of them that we're making enough ATP to keep the cell happy and functional. But in cases of stress, we get more cytochrome C oxidase molecules bound to nitric oxide than we do oxygen. And so we turn those molecules off, we're not generating ATP. But electrons are still being transported down through that chain that we talked about, And we're generating more and more oxidative stress because we're not getting to that end point of ATP generation. So what happens when cytochrome C oxidase absorbs the light is it basically pops off that nitric oxide. When the nitric oxide is released, oxygen combined, and will make more ATP. And that starts putting the mitochondria back in function, the cell back to health as we generate that energy. And that's why one of the first things we see with photobiomodulation is in fact an increase in ATP synthesis. It's the direct consequence of the cytochrome C oxidase absorbing the light, removing that nitric oxide molecule, and allowing oxygen to bind so that process can occur. Now, that little puff of nitric oxide then that's being released from the cytochrome oxidase can go into the cell and perform its other functions. Oh. And it's that puff of nitric oxide that basically sets in the longer term effects that we see with photobiomodulation, because it's the signal that's going from the mitochondria that says something's changing, transported through a series of reactions to the nucleus that says we need to change our protein expression profile.
Lovely, lovely. If I could just, to make this kind of an emotion, a in motion caricature for people, is that Professor Francisco Gonzalez Lima explained it as a catcher's mitt. So the cytochrome C oxidase will catch the oxygen like a mitt, and it kind of looks like it there. And in order to do that, though, you have to kick that nitrogen off so it can accept it. And then it pitches the ball down, the oxygen species will pitch it away, and then it catches it again and pitches it away. It catches it again, back and forth. And this is the process. So that transcription factors and things that you're talking about that nitric oxide being released, or the nitrogen ion being released, is a very key component. And we haven't really addressed it yet, so thank you for that.
Absolutely. Yeah, and I love that analogy. That's exactly what it is. It's which ball is in the catcher's mitt? Is it the oxygen ball or is it the nitric oxide ball?
Gotcha.
And they both can't be there at the same time because it's the same catcher's mitt.
Gotcha.
All right. So this figure really illustrates a lot of, a key point of why we think it's cytochrome C oxidase, and also why red light. That's important to this. So if we take a look at our graph, the closed dark circles along the bottom is what we term the cytochrome, we term the absorption spectrum of cytochrome C oxidase. It's these wavelengths of light that the molecule itself is able to absorb. Okay? Now, what we see is down around 630, 640, 670, we have a peak of absorption by the cytochrome C oxidase molecule. Then we get to a point around 720, 730, where very little light is being absorbed. And then again, as we get through 800, 900, we get another broader peak of where light is being absorbed. Now, if we think about which wavelengths of light are active in photobiomodulation, it's wavelengths 640 to 670, 630 to 670, which are this first peak of absorption of light by cytochrome C oxidase. We don't see much activity then around that 700 or 730 because the molecule can't absorb that light. And so we can't get that exchange of nitric oxide for nitrogen. But then as we get 810, 830 into 900, again, the molecule is able to absorb the light and we can set these biological processes in motion. Now, if we take a look at the red lines and the blue lines at top, this is really illustrating this in a cellular mechanism kind of way. So these cells, these were cortical neurons, cell culture, that were absorbed with light. And the red boxes are ATP content. So remember, one of the consequences of photobiomodulation is increased ATP generation. So if we look at down around that 640, 670, we have a peak of ATP generation, which then decreases at about that 700 and then increases again as we move through our 800 to 900 spectrum. And similarly, the activity of the enzyme itself follows the same curve with the exposure to the light. And so this couples for us the absorption of the light by the cytochrome C oxidase to the biological responses that we see in response to the light. All right. So I have touched upon a couple of times this concept of signal transduction. And very important to fully understanding how photobiomodulation does what it does. So under normal circumstances, the cell senses something from the external, the microenvironment, and this is detected by, say, a molecule on the cell surface, but that molecule can't enter the cell. Instead, a series of biochemical reactions basically translates that signal, transduces that signal through the cell to the nucleus, where we're able to activate gene transcription and make new proteins to respond to whatever that stimulus was. With photobiomodulation, it's a very similar process in that instead of being absorbed by the cell surface, the light is absorbed by the mitochondria. As we just talked about, the nitric oxide is released, and because this is a very small molecule, it readily diffuses through the cell. So the nitric oxide can leave the mitochondria, go out to the cytoplasm, initiate a series of analogous biochemical reactions that then translate that to the nucleus to allow protein expression to occur. And it's that protein expression in response to that release of nitric oxide that sets in place the longer term consequences of photobiomodulation that we see as wound healing, for example.
Epigenetics.
Epigenetics, to a certain extent. Epigenetics is the non-coding parts of the nucleic acid, or the DNA, if you will, that's important in regulating gene transcription, as well as other processes, but certainly it has a role in regulating signal or gene translation and transcription as well. All right.
Encoding for new proteins. This is exactly what we're going to talk about right here. Keep going. I like
right. Yep. Yep. So when we think about how a cell responds, I've talked about gene expression, I've talked about protein production, but what does that really mean? And so when we think about how a cell produces a protein, we have to start in the nucleus. We have to start with the DNA. So the DNA is our genetic material, and it contains all of the information that we need to produce different proteins. But think of it as in a quiescent or a silent state. In order for the protein to be produced, it first needs to be, the DNA needs to be translated into what we term messenger RNA. So RNA and DNA are related. They're both nucleic acids, but they're a little bit different. Whereas DNA stores the information that we need, when we translate it into mRNA, it's actually the template upon which we can make our new proteins. So as the messenger RNA is produced in the nucleus, it's modified, transported to the cytoplasm, and then it interacts with the ribosomes to make the new proteins that we need for cellular function. So when we talk about gene expression, we're talking about that stepwise process that takes us from DNA to RNA to protein. And it's the protein that's the biologically active molecule that will then elicit the myriad of responses that we that we think of as in terms of physiology. Now, when we look at what genes are turned on, what proteins do we get expressed in a cell following photobiomodulation, it runs the gambit from sodium and ion channels to cytochrome itself, molecules important in controlling that oxidative stress like peroxyredoxins. We have other molecules that are important for maintaining cell growth or signal transduction, for example. And so when we consider all of the proteins that we can see changes in a cell in response to light, it really encompasses all of the biological responses that we see as an outcome of photobiomodulation. Now, what's really important to keep in mind is you'll hear often cytochrome C oxidase is the photoacceptor for near red or far red, near red or red light. And there's no doubt that cytochrome C oxidase can do this. But when we consider the response that we see from a cell in a tissue culture dish to the response we see when we treat an animal or a person with red light, we see a much broader sense of responses in the whole organism than we do in the cell. And part of that reason is whereas cytochrome C oxidase is a photoacceptor for red to near infrared light, it's probably not the only photoacceptor in the intact organism. And that's what this figure here is to show. For example, hemoglobin, oxygenated or deoxygenated hemoglobin also absorbs light in similar wavelengths as does our cytochrome C oxidase. You'll see if you look at the dotted red line, this is oxyhemoglobin or hemoglobin bound to oxygen. The purple line is deoxyhemoglobin or hemoglobin devoid of oxygen. And depending on whether or not oxygen is present, it absorbs light throughout the same window, again, peaking at right about 600, having a nadir at 700, and then coming back up at 800, indicating that hemoglobin, whether oxygenated or deoxygenated, is also going to absorb light and probably also have a biological significance to that absorption. Melanin, the pigment in the skin, also can absorb light and therefore in the same window, and therefore will also have some biological response to that light. Likewise, myoglobin in muscle cells could explain some of the benefits that we see of photobiomodulation on muscle function. I can't hear you.
These other photoacceptors are going to be elucidated in the future, I believe, right? I mean, they're going to talk about what these mechanisms are and things like that. And
go on, I'm sorry.
Because we're going to have some other talks that are going to speak about the blood being able to, how that relates when you irradiate, if you will, the blood, both oxygen and dioxide. We talked about water and how that can be activated. Yep. No one's really talked about the melanin. And it does make sense because melanin is on the surface of your skin and the red light kind of goes to the surface of your skin. So it kind of sort of makes sense. But the benefits and the downstream effects of these are just, I mean, really starting to be expressed, correct?
Exactly, exactly. All right. So this brings us to a very important question. We know the wavelength of light is important, but we also have to concern ourselves with dose, because in this context, light is acting as a drug. And as with any drug, there is a dose. There is a beneficial dose. There is a detrimental dose. The difference between a dose of light versus the dose of a conventional drug is there are a lot of different factors that affect how much energy or how much light the cell will absorb. So if we consider all of the parameters that we need to take into account it illustrates some of the controversy that we see in the field of photobiomodulation when we discuss dose and what an appropriate dose is. We've already touched on wavelength. For photobiomodulation, we're talking either visible red light in the wavelengths between 400 and 700 nanometers, or visible light 400 to 700 nanometers, or in terms of red light, the visible or the infrared between 630 and that should be 1100 nanometers. Apologize. But we also have to take into account other measures of energy. For example, power. How much power is being administered? We talk in terms of watts when we talk power, and for photobiomodulation, typically we're administering between 1 and 500 milliwatts. A milliwatt is 1/1000th of a watt. The amount of time that we deliver energy is also important. We know that we have to deliver enough energy over enough time in order to elicit a biological response. If we flood the system with a very high dose of energy, but we only do it for a second or a millisecond, there's not enough time for the biological responses to be initiated, and therefore nothing will happen. And so making sure we're administering that light over a significant enough period of time such that those initial processes, the absorption of the light by the photoacceptor, the initiation of signal transduction, that those processes can occur so that the downstream effects can happen. When we consider power and time, this gives us our energy, or our joules. Quite simply, joules is watts times time. But we also have to be considered with the surface area over which we are administering that light. We talk in terms of energy density or power density, also referred to as irradiance. And energy density, joules per centimeter squared, for photobiomodulation, typically between between 0.04 and 50 joules. Irradiance or power, typically between 5 to 50 milliwatts. And then finally, the treatment interval. Are we doing this over hours? Is it a single dose over a few moments? Are we administering a dose every day? Are we administering a dose over weeks or months? Because the important thing is, whereas the dose that you are delivering at any one time is important, so is the cumulative dose over time. So in other words, each subsequent dose adds on to the second dose. And so you have to be concerned with not only how much light are you administering at any one given time, but over an extended period of time, how many doses are you delivering?
I think that's clinically so important, and to understand it, that is a cumulative effect, you know.
Right. Well, and we saw this in our own mouse experiments. When we started our first mouse experiments, there was a fair amount known about what was the effect of a single dose of light. We were giving our light every day over the period of a week, for example, and some of the longer term effects we saw differed from what was published in the literature. And we had to explain that it really is that cumulative repeated dose of light that's affecting the long-term outcome in our system.
You're gonna definitely touch on that because it's true.
Mm-hmm. Mm-hmm. So when we consider dose with any drug, we generate what we term a dose response curve. Basically, you have to have, how is the body responding to the amount of drug or light that's administered? And so typically what we see in a response to a drug is a dose where the dose is too low and nothing can happen because there's simply not enough drug present to elicit a response. Then we go through a period where there's an exponential increase in between the amount of drug delivered and the biological response that we see. But over time, we reach a point where we can administer more and more drug and the response is not going to improve. And there's a lot of different reasons for this. But you will reach a point where you may not be eliciting a biological response, but you may start to get toxicity. Now, when we talk about light, the important thing is it follows a dose-response curve, but it isn't our typical dose-response curve as we just discussed. Rather, it's what we term a biphasic dose-response curve. And the dose, again, determined by irradiance and time, together, joules. And again, just like with a conventional drug, we have a period where there's simply not enough light present to elicit a biological response. As we increase the amount of light that's administered, we increase our response until we get to a peak. But if we keep giving light past that peak, we actually inhibit our biological response. So if we administer too little light, nothing will happen. If we administer too much light, nothing will happen because we've inhibited the response. The exact right amount of light will activate a biological response. Now, interestingly, in terms of photobiomodulation, although there's a lot of different doses that are effective, this stimulatory dose, if you will, is typically between, say, 5 and 10 joules per centimeter squared. The other important thing to note is we make use of this dose-response curve to determine or to govern the type of response that we want to see. Sometimes we want to stimulate a response. We want to stimulate gene transcription to downregulate inflammation, for example. There are other times, though, where really what we want to do is inhibit a biological response, for example, with pain. And so when we want to treat pain, we are delivering a higher dose than so that we're inhibiting signal transduction, leading to the perception of pain, rather than stimulating something that's taking away that pain.
And at the same time, there's levels of the photodynamic that are killing cells, or high intensity lasers can take a tooth out, can do without destroy tissues. Then they can do surgery and they can do everything. So that's it's a powerful ends of each of the spectrum.
Yes. Well, and you raise a very good point, because with photobiomodulation, we're talking about low power. We're talking, as you saw in the last table, 5 milliwatts or 50 milliwatts, whereas the higher power lasers, the surgical lasers, the lasers that are cutting tissue or destroying cells, are very high power lasers. And so again, in balancing the dose of photobiomodulation, that's where balancing the power so that we can regulate cell function, stimulate cell function without inducing cell damage. And that's what the high power lasers will do. That's how the surgical lasers work. They're such high power that they're cutting through the cells, as opposed to the photobiomodulation, which is a very low level of power that can then act to stimulate gene transcription.
Oh. There we are.
All right.
Goldilocks effect, the Goldilocks effect.
Exactly, the Goldilocks zone. So again, here, the two shaded areas, the first shaded area, too little dose to elicit a response. The shaded area at the top, too much dose. We're inhibiting a response, as opposed to, as you just said, the Goldilocks zone, which gives us our perfect stimulatory response. Now, Another aspect of dose that we need to take into account is where do we want the light to reach? Now, it's important to say here that when we talk about photobiomodulation, there's a discussion of targeted effects versus systemic effects. I believe both are important based on my own work and the work of others. But if you want to reach a deep tissue site, now I should say, red light will penetrate about 2 centimeters into the body, okay? Which gets us through the skin, but it isn't going to reach deep tissue. And if we're doing transcranial, for example, we have to be concerned with making it through the skull. And so when we talk about targeted effects, we have to be concerned with the penetrance of the light, or how deep does the light go into the tissue. And this depends on the wavelength. And so when we talk about or consider the lower end of the active wavelengths of photobiomodulation, those between 630 and 670, for example, you see that if we're only talking about the skin, we transmit a fair amount of light at 670. We lose a little bit of that if we're going through just muscle, for example. And if we put the two together, the amount of light that penetrates is significantly attenuated. But as we increase in our wavelengths, and if we look up here more towards that 800 to 900 wavelength, so these are longer wavelengths, and they're able to penetrate deeper into the body and through more tissue, as is illustrated through our graph here. Again, look how much more light is transmitted through both, well, here's just skin or just muscle, but even through muscle and skin together. And so when we're concerned with going deeper into the body for those targeted effects, that's where the longer wavelengths of light become particularly important, because those are the ones that can penetrate deeper into the body to elicit those targeted effects.
Now, we've used a lot of terminology when we're talking about this, and so you're saying that in this one is that if you give if you look at a flap of skin, it's going to go through so much. If you add skin and if you add muscle, if you put a flap of muscle, it's going to go through so much. And if you add skin and muscle, it's going to go through less. Correct. Same thing we find if you just do the skull, then you do the skull and the dura and the skull and the CSF and everything like that. Now, the term that we've been using, you used it targeted, and what was the other word that you used? Systemic.
Systemic.
Correct. But I think that we can talk, maybe a terminology should be direct effect and indirect, because we've been talking about target and remote, target and systemic, and the abscopal effect means off target. So instead of using all these different words, maybe we can say direct, because we're trying to, like I say, direct meaning we're trying to influence this tissue.
Sure.
Okay? We're trying to, because we're measuring what it does, like saying a cell culture to this tissue. And so we can say this is the direct effect that we measured. But the indirect effect means everything else that they got light to it. So so that's kind of, so maybe the indirect will have systemic or remote effects, but the direct is something that we'll find right where we're measuring or looking at.
Correct. But also think of systemic in that really when we consider systemic, we're hitting the blood. We're hitting the red cells. We're hitting the immune response that's circulating. And so, as I said a moment ago, hemoglobin can absorb red light, and so tissue oxygenation, for example, as hemoglobin is absorbing light. Regulation of the immune response as it's circulating through the body to sites of inflammation, that's also part of the systemic response.
And that's one of the things that we're talking about when we're talking about looking at a brain, transcranially, say, well, this changed, the direct response was cognition improved. That's good.
Right.
But we can't talk about that with all the indirect stuff, that the blood and the skin and all the things that we just talked about. So we study one thing, and that would be the direct response, but the indirect response is the other, all the other stuff that we're talking about here. And this was, I think, was a good example.
Right. And also keeping in mind that what we define as direct versus indirect is dependent on the response we're measuring or the response we're trying to elicit.
That's correct.
So, all right. Now, also important, and this affects dose, is the pigmentation of the skin. We know that fair skin, lighter color skin allows more light to be transmitted than darker color skin. And this is going to affect the dose that's administered to our patient based on what skin we're going through. Are we going through hair? Are we shaving a site, for example? And so just keeping in mind that as we gain a better and better understanding of what dose we need to administer, that dose may need to be adjusted based on the pigmentation that the light is having to pass through. Okay, so we've talked about a lot of basic biology, a little bit of chemistry thrown in for good measure. All of this important in determining what is the biological effect of photobiomodulation. And let me say, first of all, as you're all well aware, there are many effects of photobiomodulation on the body. But when we consider at a basic level what's happening, largely, not entirely, first of all, we are modifying or modulating mitochondrial function, and we're modulating the immune response. Collectively together, these two factors, mitochondrial function and immune function, are really what is determining inflammation in the body. And as we learn more and more about disease, about aging, neurodegenerative diseases, other changes, we're finding that at the heart of so many of these pathologies is inflammation, is dysregulated mitochondrial function as well as dysregulated immune function. And this is where photobiomodulation can really have an impact. So when we talk about inflammation, we talk about pro-inflammatory or factors that are going to promote or induce inflammation versus anti-inflammatory, or those factors that are going to guard against or downregulate inflammation. So when we talk about cytokines in the immune response, there are certain factors that are pro-inflammatory. Certainly nitric oxide is pro-inflammatory by very definition. Cytokines like interferon gamma or tumor necrosis factor alpha are pro-inflammatory. On the flip side, the antioxidants that control oxidative stress, as well as cytokines like interleukin-4 or interleukin-10, are anti-inflammatory. And so when we look at pathology or even biology, for example, we're looking at a balance between these two factors or these two families of factors, and they're both important. Now, as I said at the beginning, all inflammation is not bad. We need inflammation to defend ourselves against infection. Problems arise when inflammation is not controlled, and that's where we get our pathology. So when we consider inflammation, we consider acute versus chronic. Acute inflammation is beneficial inflammation. Acute inflammation happens when we have a stimulus, say a bacteria, that enters the body. We get our macrophages, our neutrophils, that innate immunity that we talked about a few moments ago, come in first to start clearing away the infectious agent. This activates what we term our adaptive immune response, and together they're able to combat that stimulus. It goes away, and the inflammation is removed or downregulated. Chronic inflammation occurs when that stimulus is not controlled or is not removed, and basically we get caught in a feed-forward loop. And when we cannot downregulate inflammation and it becomes chronic, that's when we start to see all of the pathology that we get associated, for example, with chronic wounds, with neurodegenerative conditions. Neurodegenerative conditions, multiple sclerosis is a chronic inflammation in the body. And this is where photobiomodulation can act. Now, when we think about chronic inflammation, we certainly think of wounds and wound healing, but the important thing to keep in mind is there's a component of chronic inflammation to most of the chronic diseases, if not all of the chronic diseases that we encounter. Type 2 diabetes, for example, or cardiovascular disease are consequences of chronic inflammation, as are many neurological diseases, for example. So when we consider the regulation of inflammation, it's again the balance between pro-inflammatory molecules and anti-inflammatory molecules. And when we consider the biological effects of photobiomodulation, generally speaking, what we see is a downregulation of the pro-inflammatory and an upregulation of the anti-inflammatory. And when we consider how these molecules are regulated, it's important to note that they really feed back on each other. So if inflammatory molecules are too high, they're going to prevent the anti-inflammatory molecules from being generated or active. So as photobiomodulation comes in to tip this balance and the anti-inflammatory molecules come up, they're able to actively feed forward to downregulate the pro-inflammatory molecules. Which really, when we consider the protective mechanisms of photobiomodulation, that's really where a lot of our effects are. So in summary, as we talked about, photobiomodulation will increase ATP production. More energy allows the cells to function better. It allows the cells to defend itself against whatever insult is occurring. We're going to see an increase in anti-inflammatory factors, things like interleukin 10 or interleukin 4. Superoxide dismutase, or SOD, is an enzyme that basically removes the toxicity of reactive oxygen species oxidative stress. The flip side is a decrease in our pro-inflammatory factors, TNF-alpha or interferon gamma. Remember, INOS is that inducible nitric oxide synthase that's responsible for that big burst of nitric oxide that we get with bacterial infection or chronic inflammation for that matter. And then collectively, as we increase the beneficial, decrease the detrimental, we get a decrease in apoptosis, or that cell death, that helps restore the system to health. So when we consider the clinical applications of photobiomodulation therapy, this is, of course, not an exhaustive list, but certainly some of the more prevalent applications today. As you're all aware, recently, I should say, approved as a first line of defense against oral mucositis in chemotherapy patients, having just an incredibly beneficial effect on the treatment and outcomes for cancer patients on chemotherapy. Great promise in chronic wound healing, both pressure ulcers as well as diabetic wound ulcers. Notoriously difficult to heal, if they ever heal. And we have evidence now that with photobiomodulation, we can heal chronic wounds and prevent amputation, for example. In terms of the opioid crisis, photobiomodulation is proving very beneficial in pain management. It's able to, in a non-narcotic way, control pain. We also know that it is beneficial to treating various forms of blindness, diabetic retinopathy, age macular degeneration. Um, for a few years now, there have been a number of studies ongoing about the application of photobiomodulation to muscle fatigue or muscle function. And initially, these studies were performed in athletes, for trained athletes, and we're able to see that photobiomodulation was able to speed recovery and prevent injury, improve performance. As this has moved further down, we also now see a beneficial effect for treating genetic muscle diseases such as muscular dystrophy. It's able to have a beneficial effect. My own recent work in multiple sclerosis patients shows that photobiomodulation is able to increase strength and endurance in MS patients. Muscle fatigue, general fatigue, a serious concern for MS patients, and if we can control that symptom, we can improve their quality of life. Likewise, autoimmunity and neurodegeneration. What these things have in common is that need to downregulate chronic inflammation, chronic oxidative stress.
Can I just touch on these for 1 second? Because
Sure.
you covered a lot on this slide. So the papers that come out now on the oral mucositis. Now, is that when someone, that's the sores in your mouth that are so great that you can't eat?
Correct.
And is that with people who have chemotherapy, or is that people who have oral cancer and they have the radiation of their gum, of their tissue, of their face?
No, it isn't just oral cancers. It actually very effective in a lot of pediatric cancers. And the thing to remember with chemotherapy and radiation therapy, but chemotherapy simply targets rapidly dividing cells. And cancer cells, by definition, are rapidly dividing. However, the oral mucosa, for example, our intestinal mucosa, other sites, our hair follicles, for example, also are rapidly dividing cells. And it's why with chemotherapy, for example, patients will lose their hair because the chemotherapy is also damaging the follicles, not to a point where it's not recoverable. The hair will grow back once the chemotherapy is released or removed rather. But again, the same thing with the oral mucositis. The chemotherapy will target the oral mucosa or affect the oral mucosa, and that's where the open sores through the mouth and throat come, which just make it too painful to eat. And so not only is the body being assaulted by the drugs, it's also being
ability to reproduce itself.
Right, exactly. The nutrients that the body needs to help fight are also being removed. And so by treating the oral mucositis, and the important thing here is you can pre-treat. You can prevent the sores from even ever happening, as opposed to waiting for them to happen and then treat them. And then, so by treating or preventing the oral mucositis, the patients are able to better maintain their strength, which gives them better outcomes for the treatment of their cancer.
And thank you for that clarification, because it's just got to be known that the cellular protective or the bioprotective, not just the neuroprotective effects. And the chronic wound healing, you can also say that it's being used for not have scars, even surgical healing. Think normally.
Absolutely.
Normal healing. And, we just wanted to point that out, that the muscle function, it can take healthy people and make them super strong, supersize their mitochondria, make it bigger, so not just disease, but increased wellness, increased function. That's another thing that, you talked about. So not just disease, but also.
Well, the important thing to keep in mind with photobiomodulation is it will improve function to baseline. It won't push things into overdrive. But and if we think of compromised system with whatever that means. Now, if you're an athlete and you've just run a marathon, your muscles are compromised. You're not sick, but your muscles are damaged. Photobiomodulation can help. Everybody, I don't know this for a fact, but safe to say most people on a Western diet are going to have some amount of inflammation. Photobiomodulation is going to be able to help with that, with controlling that just kind of everyday inflammation. Right. So it really is reinstating homeostasis in the system to help preserve health.
Well stated.
All right. And I don't think I need to really state things here. This really just summarizes everything that we've talked about, knowing that light itself will interact with cytochrome C oxidase and other photoacceptors to produce the changes in gene expression that lead to the biological effects that we attribute to photobiomodulation. And then as a therapy, as we understand the mechanisms, as we develop treatment protocols, particularly effective in treating a number of biological conditions, not because it's some sort of, it is maybe a super drug in some respects, but it's active in so many things because it's targeting the very basic level of function that lies at the heart of the disease states that we study or treat.
I agree in the clinical. So theory, evidence, and practice, we kind of touched on a bunch of them and the parameters that we need. As we wind this down, you've taken us on a magnificent walkthrough of not only the last 20 years of your life, but the science that you've produced and others have produced. I want to turn around and look 10 years in the future and use your perspective to tell me, what do you expect in the field of work on how it relates to the public for 2030, 10 years from now? This is the 2020 brain and transcranial photobiomodulation. What do you see when you look forward?
I think it's an incredibly exciting time for photobiomodulation and photobiomodulation therapy. I feel honored to a certain extent to have been involved with the field for the last 10 years. We have learned so much about photobiomodulation and how it works in the last 10 years. I think the field itself is really at a tipping point. We're getting well-designed, critically thought out clinical trials that are demonstrating the therapeutic potential. We're understanding at a level we didn't previously the role of dose and how to administer dose. We're seeing more and more application, evidence-based application of photobiomodulation therapy in the clinical setting. And I think that in the next 10 years, we're going to see more acceptance in the community, in the medical community, for using photobiomodulation for pain management, for treating or preventing blindness, for chronic wound healing, for example. I think we're at a very exciting time. We just have to keep moving forward in a methodical and evidence-based way to really pave the way for this to have general acceptance in the medical community.
Thank you, Jerry Ann Lyons, for your participation. A magnificent talk. Thank you so much, and we see you soon.
Sure, my pleasure. Take care.